Ok, então vamos começar. Mesmo se está gravado, então quem conseguir ver agora, né? We will start to maintain the schedule. So our next speaker is Dr. Deborah. Just let me do a brief introduction. Deborah is a highly motivated medical doctor, statistician, and epidemiologist with over 14 years of experience in patients care and advanced medical research. Her passion lies in maternal, neonatal, child and adolescent health, machine learning, clinical trials, and bioethics. Dr. Deborah's academic journey began with a degree in medicine and surgery from the prestigious University of Ibadan, Nigeria, followed by a master in medical statistics from the University of Nairobi, Kenya. She is currently doing a PhD in tocogynecology at the Unicampi here in the medicine school, focusing the transformative potential of artificial intelligence in maternal and perinatal health under the guidance of estimated supervisors, with a resolute commitment to advancing healthcare through the integration of medicine and data-driven innovation. Dr. Awe is unwavering in her pursuit of making a significant difference in her field. Thank you very much.
Thank you so much. Boa tarde a todos. Eu vou tentar falar português pouquinho. At the beginning, just now. Depois eu vou falar inglês. So, I want to welcome everybody here once again, and I want to give a very big kudos to the organizers of this program for bringing it at this particular period of time. And I also want to thank Professor Veronica for having us here eventually at IMEC. So now we want to go to, because when we're talking about malaria, there's a tendency to focus more on the computer science part, artificial science, statistical part of it, but then it's good to bring it down to like the people population-wise, you understand, disease-wise, so we can understand a little background to infectious disease, not just about how to predict, but then we talk about the basics, so that when we talk about other things like modeling, mathematical modeling of infectious disease, which we will take tomorrow, we'll be able to understand the terminologies. Because I believe like 75% of us here are not even from health backgrounds. So I feel it's very important for us to talk about just brief introduction, because this is a whole course, like a whole year course. So, but we're going to try to fit it into 2 days. So I'll try to go slowly. Eu vou tentar falar devagar, so that all of us can follow. So if I'm too fast, você pode me chamar. Fala devagar. Okay, so now let's go into the topic. So what is the objective of this talk? Like, what's it about? Why are we talking about introduction to infectious disease epidemiology? The purpose of this course or this talk is for you to grasp the key concepts of infectious disease, and also to understand the basics of epidemiology, because we're talking about artificial intelligence to model infectious disease. So we want to talk about the other parts, okay? The infectious disease. We want you to grasp basics of epidemiology and transmission dynamics, so that when you now get into the real world to analyze and model, you see certain words, terminologies, so you can understand them and can able to, because when you don't understand the study, you don't understand the disease, it's difficult to bring reasonable analysis out of it. So you need to understand the basics of epidemiology and transmission dynamics. And also, to apply this knowledge you're going to have here to the real world scenarios. So let's go to the definition of infectious disease. So before we model, we need to understand what we are dealing with, because we have different kinds of diseases. We have diseases that are not infectious and ones that are infectious. And what is that? What makes it very tricky? Because infectious disease, the problem is that it can be transmitted from one person to another, whereas non-infectious disease, you can have it and there's no possibility of transferring it to another person. So what exactly are infectious diseases? It's an illness that is due to a specific infectious agent or the product of the agent, because sometimes it might not be the agent that causes the problem, but the product, or maybe something that the agent produces, toxic reaction to even the presence of the agent. And so this arises through transmission of that agent or its product from an infected person. So it's like, later on I'm going to talk about the epidemiological triangle, where you have a connection between an infectious agent, the person, the host. It starts, of course, with somebody that is infected, and then transmits the infectious agent within the infected person to another person that does not have the disease. So I'll go over it. It's an illness that is due to a specific infectious agent or its toxic products that arise through transmission of that agent or its product from an has the infection and then has left the room, yet you get the infection. Because you have reservoirs, maybe in some stuff, maybe the chair or anywhere the person had touched. You understand? So it can also be at a reservoir, something that keeps the infection. Is like a package that keeps the infection to a susceptible host. Now you can see that during COVID period, not everybody got COVID. So there are certain things that make somebody more likely to have the disease. That's what we call susceptible. So these terms is not used loosely. They are meaningful. I'm still going to explain what it means, because there's something within the host that makes the person susceptible to the disease. So there has to be an infectious agent that transmits it from an infected person, okay, to a susceptible host, either directly. Now, there can be direct transmission or indirect transmission through an intermediate plant or animal host, vector, or inanimate environment. I hope we understand that. Okay, so now people often mix it. Actually, can be also said to be communicable disease, but to clarify, because sometimes you see communicable disease, non-communicable diseases. Basically, communicable diseases, they are parts, they're a subset of infectious disease, and they are specifically capable of being transmitted from one person to another, or from animals to people. So let's move on to the problem of infectious disease. You see, it's a lot of problem. You see, as at 2021, we have 14 million of people dying because they had infectious disease. So it's a big problem, and that's why we need to study this trend. And you see, it's from about 2017, you have a sharp increase around the time COVID came, you see. And the problem is that we keep discovering more newer infectious disease. So I'm going to show you a cartoon before you see now. You see, this guy is saying, "Good riddance, I hope the worst is over and we can get back to normal." Then come monkeypox came. So as we are trying to overcome one infectious disease, another one is coming back. So what are infectious agents? You know, I told you, for definition of infectious disease, you need to have an infectious agent, isn't it, that is in an infected person that then is transmitted to someone that is susceptible. So we have different kinds. I don't want to go into this. If you did biology, you know we have bacteria. For tuberculosis, Mycobacterium bacterium, there's a lot of microorganisms. You have viruses. Now, why do we need to understand this? When we start talking about tomorrow, about modeling of infection, mathematical modeling of infectious disease, before you can do mathematical modeling or any other modeling, you need to understand the kind of infection, whether it's the origin of the infection, the transmission pathway of the infection, so that eventually we can know where exactly to attack to break the chain of transmission. I'm still going to talk about that. So we have all other infections also. So now the WHO has categorized infectious agents into 3. We have diseases which cause high level of mortality, like COVID, Ebola. And there are some that place a heavy burden. They don't kill. They are very gentle, but they are persistent in the environment, and they keep, gulping economic and financial, it's creates a financial burden to the country. For example, you have, in Nigeria, for example, you have malaria. It hardly kills because for a larger percentage of the community has developed some level of immunity. It still kills. It's still one of the highest causes of mortality in vulnerable population, but they don't go. You can't have a cure. So, and this affects even the epidemiological, if you see the model, you have different models for each disease condition, depending on if persons can recover or they cannot recover from the disease, or it's just a short-term thing that clears off over time. Then also you have diseases which, owing to the rapid and unexpected nature of their spread, can have serious global repercussions. For example, when Ebola came, Ebola had been endemic. All of a sudden, it caused something spill over. Things can suddenly change, something affecting the balance between animals and humans, and then maybe somebody just goes to a farm, brings it in. So it causes a lot of problem, a lot of serious, like COVID also did to us. So now, as we go on, I want to talk briefly about epidemiological terms. When we talk about epidemiology, what does it mean? I'm not going to make assumptions. That's why I'm going deep right into the basics. What is epidemiology? It's just basically the study of this distribution and determinant of health-related state or event. And then within a specified population, when you talk about epidemiology, you specify the population and you talk about the distribution. How is this disease here? For example, you have a geography. Where is it more endemic? Why is it more endemic? So you have the question, what, why, and how. So epidemiology basically is just the study within the specified population and the application of this study to control health problems. So, incidence rates is a measure of the frequency. Let me just show it here for better understanding. It's just like the proportion of new cases. You see, you open the tap. For example, you have a tap now that you use to wash your hand. The inflow is the new cases, isn't it? Then, depending on how fast the tap is running, you have like some already inside. So prevalence consists of the new cases and the old cases within the environment, whereas the outflow is maybe through death or recovery. So morbidity also is any departure from a state of physiological or psychological well-being. By tomorrow, you understand why we're talking about all of this, so you put it at the back of your mind, so when we start talking, you can follow on. Also, mortality is the number of deaths in a population due to a specific disease condition. When you talk about endemic, you see, our previous, the HOD was talking about endemic, and then you're wondering, what does it mean? It just basically means the constant presence of a disease, and it's just basically like the prevalence that I talked about. You're able to, when a disease is constantly present, it's not going, and it's just within a geographical area. Then we talk about outbreak. Am I too fast? Okay, we talk about outbreak, a localized increase in the incidence. Now, for example, like I said, at the beginning of the Ebola crisis, it was just an outbreak because it was endemic within the area. But after a while, it became an outbreak. So when a disease condition now goes beyond just a locality and then spreads maybe to more people beyond like excess of normal, that is what you call epidemic. So when does it become a pandemic? When it spreads from one geographical region to another geographical region, like from countries. That's what we call pandemic, like in COVID case, where it spreads all over the world. So now, let's move now to the epidemiological triangle. This is a very key concept, key in-depth. When we talk about epidemiology, it's like one of the major important concepts in epidemiology. Here you have the agents. I've talked about it before. You have the host, and you have the environment. So these are the three, what we call it, you see, it has three corners. We call it three agents, three participants in disease transmission. So for an infection, a disease to occur, there are some things within the agent that makes it more likely to transmit infections. And that depends on the type of organism, whether it's virus or bacteria, also on the toxicity, how virulent. There's some infections that once the person enters here, the probability that everybody has it is high. Meanwhile, like HIV, you can't contract HIV by just sitting down beside someone. So it depends on what? The virulence and the nature of the disease. And also the susceptibility to antibiotics and so other things. Then some other agents. You see, for example, like flu. Why have we not been able to eradicate flu? Even after developing vaccines. You take COVID vaccine today and tomorrow, you get cold again. Why? Because the organisms keep what? Mutating. They keep changing. You see, they add a protein to the DNA structure and then it's a new, they call it another name. You understand? So that's why it keeps escaping even vaccine. So there are things within the agent that also affect whether it's going to continue or have a sustained effect on the host. And so what are within the host? The age. You see, for babies now, if they come here, that's why they don't let babies or children enter the hospital except they are sick. Because they have a higher tendency of having the disease because of their immune system. Pregnant women. So there are things within the host that makes the host more susceptible or less susceptible to having the disease. Then prior exposure. Some people with prior exposure, they then develop immunity that makes it difficult for them to contract the disease again. Then co-infection. There's some people that they already have an infection. So the presence of that infectious agent makes it worse. So presence of co-infection within the host. Then immune response. Some hosts, they don't have enough immunity to fight a certain, because of they have other immune problems. Or they have like maybe comorbidities, like things like HIV, diabetes, HIV. That's why during COVID, you see, there were certain people that were given more preference because they are more likely to die from the disease. And they actually died. A lot of people died because of the lack of immunity. So the immune response of the host, if it is affected, it can affect how fast the host contracts the disease. And so for the environment, we have climate. Nowadays, just talked on last week. Now you have like some like flu. When you have erratic changes, you see, in weather conditions, there are so many other conditions that thrive even during the rainy seasons, some during the wet, dry seasons. So this also affects how much an agent can survive within the environment also. And then population density. For example, if we were, the more we are in this room now, I'll talk about it tomorrow, density dependent stuff. Because the more we are, the more likely you contract flu if somebody has flu. But the less, so the disease is dependent on the number of people, the density. That is the quantity of people within this place. For HIV, whether you are more or fewer, it doesn't affect that it's going to be you're going to get it. So there is population density, then social structure, and so on and so forth. So I've talked about all of them. Let's move on to host immune response. I've said earlier on that the immunity of the host affects whether the host gets the infection or not. And so the immune response is the body's defense. What is it? What does it mean? It's the body's defense mechanism against foreign substances, such as pathogens, and then some bacteria, fungi, and then other harmful agents. And it involves a complex network of cells, tissues, and molecules working together. You see, they recognize the disease, and then the body stimulates like an antibody reaction to the presence of the agent. So with time, we call something immunity. See, I'm not saying everything in details, but pointing to areas that you need to keep in mind as we move on in this particular aspect. So immunity to a disease is conferred by presence of antibodies in the individual systems. And the antibodies, of course, we know that they are proteins synthesized by the immune system to eliminate toxins and pathogenic organisms. So these are the terms you are going to be seeing tomorrow. Susceptible, okay? Key terms to describe an host. There are different people here. We have different set of people. We have susceptible ones, that is, they are uninfected. Keep in mind, they don't have infection, but they are able to become infected, okay, if they are exposed. Number two, we have the infectious. There are people that are already infected and they're able to transmit the infection to people that are susceptible to the infection. Then we have people that are immune to the disease. They have cell-mediated or immunity against the disease, okay? And it protects them against the infection. That's when you say someone is immune. Then diseased or clinical infection. These are the people, the presence of clinical signs and symptoms. The person is already coughing, is already wheezing, you have other symptoms, add fever. Those are the people that have this, they are already diseased. Then we talk about latent infection or subclinical infection. Some people, they might be diseased, but they don't show symptoms. They're already diseased, they have the infection, it's already working on them, but they don't really show clinical symptoms. And that was why COVID was able to spread so fast, because some people would carry it for a certain period of days before they start showing symptoms. They're already diseased, but it's not showing outside. So that's what we call latent infection or subclinical infection. That is the presence of infectious agent but absence of clinical disease. And so the person can still carry the disease but does not show symptoms. Then we have the carrier. These are the people that have protected infected state. Usually we call them chronic states. For example, hepatitis A is caused by hepatitis virus, but it does not have a chronic state, unlike hepatitis B, where when you have it, you become a chronic carrier, like HIV. It doesn't go. You can only maybe reduce your viral load or increase your CD4 count, but the person becomes a lifelong carrier. So it implies a protected infected state with shedding of infectious agents. The person is infectious, and carriers may be diseased, recovering, or healthy. So now, let's talk about more terms. Because when you talk about infectious disease modeling, you will start seeing some models. You see susceptible, exposed, infected. So basically, the structure of the disease itself, the infectious disease, the nature of the disease affects a lot of things. And then the host itself, what goes on in the host, also affect the transmission of disease. So we have some people that are exposed. That is, they already have come in contact with the pathogen through maybe inhalation, maybe ingestion, or just physical contact. These people are exposed. Then we have the infected. You see, those are the largest proportion. You can look at the circle here. You have the infected. Those are the people that the bacteria or the virus has entered and then begin to multiply. Okay, then we have the infectious. These are people that can transmit the pathogens. Basically like the repetition of what I've said before. But don't forget this graph. So now, the disease progression. I've talked about latent period. For more emphasis, that is the time between when you have contact with the pathogen and when you become infectious. This is at the point where this person becomes infectious. This is the infection, the organism. Then, onset of shedding. Okay? There is no clinical symptom. It's there from incubation period, where is the distance between the infection and the onset of clinical symptoms. So when you talk about latent phase or subclinical phase, this is what? The distance between infectious agent and the what? The time it takes after you have been infected with the disease, and then you start shedding the virus, maybe through the blood or any other place. Those incubation period I've said, the first symptom you have of the disease, that is the incubation. How do they calculate? You'll be wondering, what's the incubation period of COVID, of this? They calculate it by noting when you come in contact with the infectious agent and the first sign of that disease in the body. Then we have the what? We have the onset of shedding. I've talked about it, when you start excreting the virus or the bacteria. Then infectious period is the time which an infected individual can transmit the pathogen. Are we okay with that? Then we move on to what? The clinical disease, or already I've showed you before, that's when you start showing signs of and symptoms of illness. All these terminologies are very important because when you go into the full details of infectious disease modeling using mathematical models, you will need all this to be able to help you. So serial interval is defined what? What is it? Is the time between the appearance of symptom of the previous infected person and the newly infected person. The distance between the onset of symptom for both. When the first person gets the symptoms and the other person also starts showing symptoms of disease. So, what are the ways how are disease transmitted? Disease transmission can be direct or indirect. Well, for example, through mucous membranes or like body fluids, like blood. You see, this is what sexually transmitted infections. Then you have across the placenta from the mother to the child, for toxoplasmosis and some other touches, they call it touches. Then transplants also. Then skin to skin, like herpes. Then chickenpox also. So chickenpox can be transmitted both through direct contact and indirect transmission. Then influenza, when you sneeze, those are direct transmission. Indirect transmission is like you need to have like an interface between the host and the other host. It's not directly transmitted, basically. Waterborne, for example, the disease is within the water, and then you take the water. That's indirect transmission, for example, hepatitis A. And then chickenpox also can be transmitted indirectly because, for example, the clothes that the person is wearing, when you have contact, you can get it also. So it can also be transmitted through other things. Then foodborne, maybe when you eat the food, and then the vectors. That's basically what I'm explaining on this other side of you see, by food, by insects, and so on. Why do we need to understand disease transmission? We need to know this because when we start writing the models, we need to understand how it is transmitted in order to be able to know how to model, because everything is going to be included in the calculations, okay, for the mathematical model. So we now go to malaria. I just wanted to talk briefly because the emphasis of this particular workshop is on malaria. So, but I don't want to go into details. It's basically the transmission of malaria. You see from the host, the initial host. This is the initial. Mosquito is already infected. Okay, it then bites the susceptible host. The whatever it releases inside then matures, enters inside the liver, multiplies, or it can be transmitted to the unborn child. Then you have the release of the sporozoites into the blood system, and then another mosquito, healthy mosquito, bites the human being and then sucks the blood, then transmits to the next one when it bites it. So this is basically, what kind of transmission is this? Direct or indirect? Indirect. Thank you. So because it's not, you can't contact malaria by sitting beside someone. So that has to be a vector. Okay, that's indirect transmission. Just like dengue fever is the same. It's basically the same, not the same organism, but the same mechanism of, mosquito-borne disease. So for coronavirus, I just want to talk about the transmission, different transmission dynamics. You see, for infected individual, you have the droplet, maybe when you cough or you sneeze, and then it goes to another individual, and so on and so forth. So now I want to talk about chain of transmission. I'm about to end this particular presentation. We talk about the chain of transmission. That is basically like 5 parts. I talked about the epidemiological triangle. That's 3: agent, host, and what? What is under it? That's environment. So for this chain of reaction, we have what? It starts with what? You have the susceptible host that gets infected with the bacteria. Okay, then it goes into a reservoir. I've already talked about some of this, like the environment. Okay, then there's a place exit point. Let me go about susceptible host. I talked about whether young or old. Then the organism. Then the reservoir. It can be the food, human, animals, and so on. Then there is a port of exit. It can be through the saliva or urine. Then the transmission, whether through contact or droplet or airborne or vector or fomite. Then it enters again through where? Mucous lining. So basically for chain of transmission, when you hear something like this chain of transmission, it consists of what? A susceptible host, the microorganism, the reservoir, port of exit, modes of transport, and the port of entry. Is it boring? You know, medicine is very interesting, but sometimes epidemiology can be boring. So I'm going to round up very soon. So this is basically what you're enjoying. Oh, great. So that's basically the chain of infection. And then this is just a diagram of it when you cough and then basically repeating the same thing. So now the goal of epidemiologists is to break this chain, to look for areas around these chains and then look at ways of truncating. You see, like the HOD said earlier on, he said that concerning infection, what did, I tried to remember what he said concerning, no, let me go back to breaking the chain of infection. So you look for areas where you can truncate, you see, like this, to stop the flow of. Now, yes, I've remembered what I wanted to say. When you talk about epidemiology, you see, doctors, like 40% of times, he says 40% of times you can be right. But then what infectious is, what machine learning does is to increase, the accuracy that can't be done by human, because we have human error, which can be intrinsic or just maybe by reading something wrong. You can make a wrong diagnosis as a doctor. But machine learning, as he has said, helps us more. But then I'm talking about now the epidemiological way of preventing disease. I'm not talking about how machine learning helps us to predict. So now, what are the things? For example, for communicable diseases, we have disease caused by an infectious agent. Preventive methods that we use includes pasteurization, then chlorination. For example, water, you can add chlorine to break this transmission. You see the reservoir, it breaks this point. Okay? That point that connects the microorganism to the human, it breaks it along this point by what? Disinfectants. For example, somebody has come here, you say, what? You sanitize your hand. Or you sanitize the environment. That's why in the hospital you see continuous cleaning. So that it's not just because they want to keep it clean, but just to also take away, wipe off any form of infection. Then also the human reservoir that I talked about, the reservoir here. What do? We can do isolation. That was what was done during COVID. So to break that connection, the link between the host and another susceptible host, you remove that diseased host. So we do isolation, we do surveillance to see areas where you have more disease. Then we treat, because sometimes when you treat, you're able to clear the disease. Not all diseases can be cleared by just treatment. So, and then we have portal of exit, this point. We have secretion, saliva, urine, and so on. What do? We utilize handkerchiefs, condoms, for example, like sexually transmitted infections. Okay, the host is already diseased. How do you prevent another host from getting it? So you do what? You block the exit point, portal of exit. Okay? And then ear nets, insect repellents. You try to, hand washing, okay? Those are areas where you can have secretion. For example, hepatitis B, you can get it from body fluids, not just from sex. You can also get it through body fluids, and so on. Sweat. Kissing. Or like HIV, that the person has to have a wound in the mouth for you to get it through kissing. So all these parts you need to also know about. Then the transmission. We do what? Isolate and washing, and then we do sexual abstinence, of course, and then some other stuff. Then for portal of entry into the host. See, this is portal of exit from the diseased person. For portal of entry for another host, a susceptible host, what? Condoms, hair nets, insect repellents, and washing. So, what are the questions? This is just an introduction to stimulate you. So as epidemiologists, what questions do we ask? We're facing, because, the world is like, for example, like 4 years ago, did we know we were going to have COVID? So because all of a sudden it can be a spillover, there can be a problem somewhere, and it just disturbs the equilibrium. So there are questions we ask, especially when we want to face an impending epidemic. And then what are the questions we ask us? What is the severity? What's going to be the severity of this disease, of this epidemic? How do we know that? We count what? The total number of individuals who need medical attention. Okay? That will tell you the burden of the disease. And then the peak number of infected individuals at any one time, at any time point, like a point prevalence. You count the number of infected people. Then number 2, we ask, what is the duration of the epidemic? This includes understanding when it will reach its peak. Those are the questions we are going to answer in the next class, okay? Like, how do we know all these things? Because if you understand the duration of the epidemic, we can plan, we can make policies to be able to address this epidemic. Then number 3, what is it effective to quarantine? And what vaccination should be done? And then how much vaccine or antiviral medication will be stored in advance. So now we are moving just beyond response to the epidemics. You see, what we do nowadays, we are moving more to preparedness. What if another epidemic strikes? Then what they are doing now, they simulate. Okay, let's say we have an epidemic. What are we going to do? So all these things help us. This knowledge will help us to be able to do what? To plan for the next epidemic. So the last one is, what strategies will be most effective for containing, controlling, and eliminating the disease? So we cannot talk about infectious diseases control or modeling, because what are we predicting? What are we even predicting? Why are we using machine learning? Why are we using artificial intelligence? We want to improve, isn't it? To improve the outcome. Is it that there are different points in time where you can also do what? Intervene. It can be maybe to prevent the disease. It can be for increased what? Management of the disease, to know the effective drugs or the vaccines to use. Then it can be also to prevent people from complications. So those are areas where we address in epidemiology or even in medical practice. So why do we model infectious disease? You know, this modeling of infectious disease started a long time ago. In fact, I got one dated as far back as 1927. Why do we do that? Because before now, we don't have much data. Why are we talking about artificial intelligence? Because we have data now. Like, we have a lot of things going on. We have a lot of data being taken. Sorry? What am I doing? Oh, he's here. Okay. So, now that we have data, that is where we can do infectious disease modeling. But then, we don't have so much data. But then, I read a study just a few minutes ago that when they did the comparison, I can share the link to the study, between mathematical modeling of infectious disease, statistical models, and artificial intelligence. Of course, AI did better, but when it comes to understanding the spread of the disease, the nature of disease, we still resort to mathematical modeling, isn't it? Because it gives more explanation about the host and the agent itself, as we will see next time. So lack of data or incomplete data is why we resorted to mathematical modeling of infectious disease, and also to understand the disease spread. How is it spread among places? You see mapping, you see a lot of network models, stochastic models. Then, number 3, refine focus. Developing a model requires us to explicitly define the assumptions, because for each model, you have to have an assumption based on what? The type of infectious disease, the mode of transmission, and the other things, whether people recover from it, because it's not every disease that can be recovered from. So because of that, you develop an assumption, as we'll see in next class. So this is just an example. Why I keep talking about susceptible, I've repeated it many times. This is the way it looks. Tomorrow we're going to talk more about compartmental models for infectious disease. So it looks like this. You have the susceptible, the exposed. This looks like a malaria or maybe dengue fever. Because why? For example, when you have susceptible people, it's not everybody that gets exposed to mosquitoes, isn't it? So you have to have an exposed part of it. For some, you have the cell model. I'll talk about it tomorrow, where there is no exposed. It's just susceptible, you become infected. Because there is nothing like exposed part of it. So this is just like to give you a brief overview of what we're going to do in the next class. This is why we model infectious disease, to be able to understand what is the infectious rate. Now we can calculate how much can this disease, how fast can you get infected? What's the force of infection? Then we can calculate the prevalence, even from here, the incubation, then the recovery rates using mathematical models. And so, meanwhile, compared to statistical modeling, what do? You get the data, then you fit it. It's different from here. Here, you calculate the number susceptible. You have to know an initial point. Then, you now calculate. If one person has a disease, how long will it take for another person to get the disease? We can actually calculate it with just small data. You just need the number of susceptible, number of exposed, number of infections, number of recovered, not specific data for individual patients. You see how it looks simple, but it's very complex. So what are the major takeaways from this conversation today? Number one, just to understand what infectious diseases are. Okay, I hope you can tell confidently what an infectious disease is and differentiate from a non-infectious disease. And then you can gain what? Comprehensive understanding of what constitutes an infectious disease, including the cause. And then the classification, like I talked about, the WHO classification. Those are those basic epidemiology. Then the key epidemiological metrics. Talked about morbidity, mortality. I've talked about incidence and prevalence, and then the death rate. So now, also to get familiar, when we start talking about epidemiological measures, you can get yourself familiar with it. You can understand latent now, compared to when somebody is really diseased. And then application of the epidemiological triad. How we use the epidemiological triangle, the agent, the host, the environment. How we use it to what? To do what? To model the disease. Okay? And then to also look for ways to cut off the link between each of them. Because once you're able to break any of the triangles, then infection might not occur, disease might not occur. Then the interplay between host, agent, and environment, the transmission dynamics. I've given an example of malaria. Then to recognize various modes of transmission, the direct and indirect transmission. Thank you so much. That's all I have to say today.
Thank you very much, Deborah. Questions, comments? Please.
For me, it's not so clear the difference between susceptible and exposed yet. So could you give another example, please?
Okay, for example, susceptible. Okay, let me just use. A susceptible individual, the person does not have the disease, but it's possible to have the disease. Somebody exposed has already come in contact with the infectious agent. You can be susceptible and you will not yet have interaction with like say mosquitoes. You can live in an endemic area, like maybe in the northern part of Brazil, and then you are not bitten by mosquitoes, you will not have the infection. So exposed are people that are exposed to the maybe mosquitoes bit you, so you're already exposed to or you had contact with somebody that had COVID. You can be at your house, you don't go anywhere, and you're not exposed to COVID. But when you go, maybe you go to a place and then you discover that your friend went to a party, and then you heard that your friend had COVID. So you are exposed. You understand? People that were not in the party were not exposed, but they are still susceptible to having the disease, because there's nothing protecting them from having the disease. Okay?
What would be an example of without exposed? Because you said you have some models that they have the susceptible part, infectious part, and recovered part without exposed. So
okay. Let's say, for example, your wife is at home, she's not exposed to the disease, but she's susceptible. But you went and had contact with somebody with the infectious agent. or you went to a place where the infectious agent was in the air. You understand? So there's still a small distinction between the susceptible and exposed. Let me go back to the definition. Maybe it will help you better. Okay.
Just to add or to clarify him, I think you can understand the susceptibility in terms of if you have a malnourished child. A malnourished child is deficient of nutrients, is deficient of so many things that could predispose him or her to any infection. So that person alone is already susceptible because he or she has an underlying immune compromised immunity that is already making the person to be susceptible. That's just how to clear that one. Then exposed, she has already done justice to that.
Thank you very much. Thank you so much. Now, another thing is, if you are not in a place, let's look at this one. For example, see, exposed refers to the state of having come into contact with a pathogen. That's all. You have to come in contact to be exposed. But you can be susceptible but not exposed to the pathogen. Susceptible just basically means that you are likely, you are uninfected but you are able to become infected. Just ability to become infected. Do you understand?
Yeah, I think the susceptible is more clear for me than the exposed.
Okay.
So I think, well, in my case,
I understand.
This definition of susceptible is very clear,
Mm-hmm.
but the definition of exposed, it's kind of clear for me. But
okay.
I think
let me just
people could ask
yes.
other questions.
Let me say it again. If you are susceptible to a disease, you can become infected by the pathogen. But when you are exposed to the disease, you already have contact with that pathogen. You can be susceptible yet not have contact with the pathogen. Do you understand? That's it.
Thank you.
Any other question?
So you said in a direct way, less flu and COVID, it's easier to not get exposed by the virus or the pathogen. But in a case like malaria, it's a mosquito. How you could stop the cycle of infection? When you stop on people, you can eliminate malaria on people, but still on the eggs of the mosquito, or if you stop on people, it will stop this infectious cycle.
The best way to stop the malaria is to, we have agreed that, thank you very much. The best way, when you talk about elimination, is not used loosely, it's close to eradication of a disease. So basically to eliminate malaria is to eliminate this. Because that's the link between this and this next human. So to eliminate malaria, you have to get rid of mosquitoes.
Getting rid of mosquitoes vector, it wouldn't affect in nature, it wouldn't affect the environment as
I understand. No, you are talking about the food genome or the food, is it bio, you're talking about the food chain. Are you talking about the food chain?
Yeah,
so it's
this vector.
Yes.
It would affect the environmental way,
No, it won't.
No?
The advantage of mosquitoes. There's no advantage.
Mm-hmm. Right.
It has no advantage. When we talk about maybe eradicating cats, can you say now, for example, you can transmit COVID? Let's say Ebola from rats.
Mm-hmm.
Can you eradicate rats? It's not possible. Is it possible? Even if you plan to eradicate rats, you keep having multiplicity. But for malaria, what they are doing now, if I get it correctly, is to genetically modify the parasite in such a way that when you get it, it doesn't really. That's what they're doing in US now to eradicate malaria completely. Yes, they genetically modify the disease. And then there's malaria vaccines too now. but the effectiveness is 50% last time I checked. So the best way to eradicate malaria is to get rid of these menace mosquitoes. Thank you so much. Any other question?
I have one. It's very fast. Sorry. I remember that during the COVID, some people used an index R0 or something like that.
Basic productive rate. I'm going to talk about it tomorrow.
Okay.
Because if I talk about it today, I'm going to forget because it's very much linked to. We're even going to do it in half. So we're going to do it tomorrow.
Because this is an indirect, not direct like in other case, but maybe something like one mosquito can contaminate 3, 4, I don't know.
So any basic reproductive rate more than 1, that means the infection has a probability of becoming an epidemic. And if it's less than 0, it's not likely to become an epidemic, but it will remain an endemic situation. So basically, basic productive, it's a formula. I'm going to talk about it tomorrow, don't worry. It's a formula, there's a way it's calculated. Everything we'll talk about it and how to calculate it. So it's between the susceptible and the exposed. How much, if you have contact, the susceptible has contact with the disease, how fast is it going to send it to another host? That's just what it's measuring. How fast will you be able to, for somebody that is infected to get to another person that is uninfected? So you calculate the ratio. Once you have the ratio, you know that, by this particular time, this epidemic, this thing would have become an epidemic. So you can actually forecast using this. So it's just a set of equations that they calculate using the force. We'll get to that. I don't want us to, or you want to look at it?
No.
I'll show you tomorrow. It's a big topic.
It's a big topic.
Yes.
It's very interesting because I think that this can be the key for prediction.
Yes. Especially when you don't have so much data. You just have very basic informations. Number of susceptible, number of infected, number of recovered. You can calculate a lot of things from that.
Thank you. Let's thank again Deborah.